I want you to imagine trying to build Noah's Ark, but instead of animals, you were building it for plants.
Right, which is quite the visual.
Yeah, like how exactly do you pack an entire forest into a freezer?
I mean, the logistical nightmare of that is hard to even wrap your head around because you aren't just taking a snapshot of a forest.
Right.
You are trying to capture the functional genetic mechanics of an entire ecosystem so it can actually be rebuilt from scratch one day if, the worst happens.
Welcome to this deep dive into the source material. Today, we are looking at the monumental high-stakes science of global plant conservation.
It's a massive topic.
It really is. And the stack of documents powering this deep dive is just incredible. We've got a Millennium Seed Bank Project field manual straight from Kew Gardens.
Such a fascinating document.
Absolutely. And we also have a Darwin Initiative report detailing efforts to save the highly threatened forests of Chile. Plus, there's a dispatch from Island Conservation on the remote Juan Fernandez Archipelago. And to top it all off, we have this highly technical scientific workshop guide on the actual literal physics of seed storage.
Yeah, and what this collection of sources gives us is the complete picture. We are looking at the entire chain of custody required to essentially cheat extinction.
And that is exactly the mission for today. We're going to demystify exactly what it takes to save a single plant species from disappearing forever.
Step by step.
Exactly. We're going to trace that journey from the muddy boots of researchers in a South American forest through the incredibly dense layers of international bureaucracy and law.
Oh, the bureaucracy is intense.
It's wild. But we'll trace it all the way to a sub-zero high-tech vault in the UK.
And this matters to everyone listening because we are talking about the ultimate backup plan for Earth's biosphere.
Right. Whether you are a casual backyard gardener or someone prepping for an environmental science meeting or just an insanely curious mind, you should care about this.
Definitely.
Because the sheer scale of this backup plan is going to blow your mind.
But I guess before we step into the sterile environment of the lab, we have to understand the ambition here and the rigorous rules of the harvest.
Yeah, and that scale is probably best captured by the Millennium Seed Bank Project, or MSBP.
Right, the Kew Gardens Project.
Exactly. Their baseline goal, which is outlined in this field manual from 2001, was to collect and conserve 10% of the world's seed-bearing flora by the year 2010.
Wow.
And they were focusing primarily on dry land.
10% of the global flora. That is what, around 24,000 species?
Roughly, yeah.
But reading through the manual, you quickly realize that you can't just, throw any seed into a deep freeze, can you?
No, not at all.
Because they divide the plant kingdom into two main categories, right? Orthodox and recalcitrant.
Yes, and that biological baseline really determines everything they do. So an orthodox seed is one that survives being dried down to a very low moisture content and then frozen.
Okay.
Those are the ones you can bank for decades or even centuries. And the recalcitrant ones.
Well, recalcitrant seeds like certain fleshy fruits, or there's that threatened Chilean conifer mentioned in our Darwin Initiative sources, the Premnopithecus endandia, they are the complete opposite.
Because they just have too much water in them.
Yes, and they actively need that water to survive. If you try to dry a recalcitrant seed, it simply dies.
Oh, wow.
And if you try to freeze it without drying it, the water inside forms ice crystals, which basically ruptures the cellular walls, and again, it dies.
So they just burst.
Pretty much. So for the purposes of these massive frozen seed banks, the focus has to remain heavily on the orthodox species.
Okay, let's unpack this. Because for the orthodox seeds, the ones they can physically bank, the collection rules in this manual are intense.
Very intense.
Like, the field collectors aren't just taking a casual stroll through the woods and picking up a few pine cones.
No.
It reads like a high-stakes genetic heist mixed with extreme accounting.
That's a really good way to put it.
Because they're instructed to gather 10,000 to 20,000 seeds per population. Why on earth do they need 20,000 seeds? Isn't a handful enough to grow a few plants?
Well, if your only goal was to grow a pretty museum specimen in a glass house, sure, a handful would be fine.
Right.
But the goal here is population survival. The mathematics of survival dictate that you have to capture the maximum genetic diversity of a wild population.
So they can handle whatever the future throws at them.
Exactly. So it can eventually be reintroduced to the wild and adapt to future threats. To do that, the manual dictates a strategy called stratified random sampling.
Stratified random sampling.
Right. From at least 50 individual plants in a given population.
And what does stratified random actually mean for a researcher who is just standing out there in the dirt?
It basically means you have to actively fight human nature.
How so?
Well, you don't just harvest from the biggest, healthiest bush right next to the road just because it's convenient.
Oh, I see. Because that would skew the genetics.
Exactly. You sample evenly across different microsites, so some plants in the heavy shade, some struggling on a rocky slope, some down near a stream.
Okay, that makes sense.
The manual actually references a 1995 paper by Brown and Marshall, which shows that by randomly sampling 50 individuals, you can capture at least one copy of 95% of the genetic alleles that occur in that population at a frequency greater than 5%.
Wait, for the non-biologists listening, what exactly do we mean by an allele here?
Good question.
Are we just talking about genetic variations, like different flavors of a specific trait?
That is a great way to think about it. Yeah, an allele is just a variant form of a gene.
Okay.
So some alleles might dictate taller stems, others might dictate a slightly deeper root system.
Hmm.
By getting 95% of these variations, you are capturing the vast majority of the genetic software of that specific population.
Because you don't know what they'll need in the future.
Right. Because you have no idea which specific flavor of a trait holds the key to surviving a future drought or a totally new pest. You need the whole library, not just one book.
That is fascinating. And the amount of data they have to collect alongside those physical seeds is heavy, too.
Oh, it's a ton of paperwork.
I was looking at the field data form, and it requires latitude and longitude from GPS, the soil pH, soil color, drainage, the land.
Modifying factors, too.
Yeah, exactly. Modifying factors like whether the area is grazed by animals or regularly burned by wildfires. They even collect ethnobotanical uses.
Yes, the cultural context is highly prioritized.
Right. They are noting down the local vernacular names and what the indigenous or local communities use the plant for, like whether they boil the leaves for a headache or use the wood for fuel.
Because a seed stripped of its environmental and cultural context is practically useless for future restoration.
Really?
Useless.
Pretty much. I mean, if you try to plant a seed 50 years from now, but you lost the paperwork saying it originally evolved to thrive in highly acidic, well-drained soil on a 30-degree north-facing slope.
Your restoration effort will probably fail.
Exactly. It will likely fail.
So we have these bags filled with 20,000 carefully sampled seeds and clipboards full of soil and cultural data. But we have a major logistical hurdle now.
Borders. Yeah.
Yeah.
You can't just toss all this genetic gold into a suitcase and catch a commercial flight back to London.
No, you definitely cannot do that. Moving genetic material across international borders is a highly regulated, heavily scrutinized process.
Right.
The Convention on Biological Diversity, often referred to as the CBD, firmly recognizes the sovereign rights of nations over their own biological resources.
Plus, you have CITES regulations, right?
Yeah.
Which control the international trade of endangered species to prevent them from being smuggled or exploited.
Exactly. You absolutely must have what is called prior informed consent before you pluck a single leaf.
And this is a massive point of friction, isn't it?
It is, because historically there is a deep, valid fear among developing nations of biopiracy.
Oh, right.
Wealthy countries sweeping in, taking native plants, synthesizing a drug or a crop from them, and basically keeping all the profits.
Which brings us to the Darwin Initiative report from Chile.
Yes.
Because this becomes a brilliant case study in how you actually negotiate that friction.
It really is a great example.
They spent years hammering out a benefit-sharing agreement. And instead of just listing the alphabet soup of institutions involved, from the Chilean government authorities to the Botanic Gardens in Edinburgh, let's look at what they actually agreed to do.
Because the deal they struck is pretty wild.
It is entirely groundbreaking. They identified 28 specific Chilean plant species that hold high potential for the UK horticultural market.
Right.
And the agreement allows the UK institutions and a commercial nursery to propagate and sell these native Chilean plants to everyday British gardeners.
But with a catch.
Right. A mandated portion of the net profits from those sales is funneled directly back into a conservation fund in Chile, which is overseen by a joint committee.
Yes.
Now, here is where it gets really interesting and where I have to push back hard.
Okay, let's hear it.
They are leveraging capitalist commercial UK gardening trends to fund wild native forest protection in South America.
They are.
Isn't putting a price tag on a threatened species a really dangerous path? Doesn't that just invite illegal harvesting or overexploitation once people realize there's money to be made?
That tension is at the absolute center of modern conservation. And honestly, the project leaders were hyper aware of the optics and the risks there.
So how did they prevent it?
Well, The legal framework has ironclad clauses prohibiting overcollection from the wild. The commercial plants sold in the UK are propagated only from the initial legally acquired samples.
Okay, so they aren't going back for more wild plants.
Exactly. But more importantly, the experts had to actively manage everyone's expectations about the money itself.
Because very few plants are actually going to be commercial blockbusters.
Precisely. They aren't going to generate millions of dollars overnight from selling some shrubs.
Right.
The true value of this benefit-sharing agreement wasn't the eventual cash flow. It was the fact that they spent the time to build institutional trust.
Oh, that makes sense.
They created a legal template, proving that foreign institutions can legally and ethically help conserve another nation's resources without stealing them.
So the treaties are signed, the trust is built. Let's look at what this protection actually looks like in the dirt of South America.
It's a very different picture on the ground.
Yeah, when you read through the Darwin Initiative report on central and southern Chile, it is pretty bleak. We aren't talking about vast, untouched wilderness here.
No, not at all.
These native temperate rainforests have been completely chopped up over the last 30 years.
They suffer from severe fragmentation. The native land has been cleared for agriculture, logged for wood chips, and largely replaced by massive, highly profitable commercial plantations of exotic pines and eucalyptus.
And the native species that are left, like the highly threatened tree Batavia punctata, are often clinging to existence in tiny, isolated fragments of forest, many of which are on private land.
Right.
I mean, to even help people realize what they're looking at, the project published a comprehensive identification manual called Plantas Amenazadas del Centro-Sur de Chile.
Which is a great resource.
It is, but identifying a rare tree doesn't save it from a chainsaw.
No, it doesn't. And that is where the Darwin Initiative's strategy became incredibly pragmatic.
What do?
They worked to secure conservation agreements with small local landowners. But significantly, they also walked right up to the massive commercial forestry companies, giants like Forestal Maninco and Forestal BioBio, and negotiated with them.
Wait, I need to pause you there.
Sure.
The conservationists are teaming up with the massive timber companies whose pine plantations literally surround and choke out the native forest fragments.
Yes.
That sounds like a textbook fox guarding the hen house situation. Like, how do you trust the logging company to save the trees?
I know, it sounds totally counterintuitive, but it requires looking critically at how fragmented landscapes function.
Okay.
If the forestry companies legally own the land where the last remaining populations of a species exist, you really have no choice. They have to be your partners.
But what is the incentive for the timber companies? Why would they slow down their logging operations to save a rare bush?
Certification.
Oh, really?
Yeah, the global market increasingly demands sustainably sourced timber, and part of getting that lucrative certification involves showing that you are actively protecting biodiversity on your land.
Ah, so it's a business advantage for them.
Exactly. But here's the catch. These companies often lack the specific botanical expertise to even identify the threatened native plants hidden in the steep gullies between their pine grids.
So they don't even know what they have.
Right. So the Darwin Project provided specialized training to the forestry staff, and in return, the companies agreed to fence off and protect the native fragments.
But does relying on commercial timber companies actually work? Like, are the plants on that private land actually doing better than the ones safely inside government national parks?
They absolutely are, and this is where the genetic research comes in. A Darwin scholar named Darian Stark proved something astounding. His work showed that the most genetically variable, distinctive, and therefore valuable populations of Batavia punctata were actually located outside the official protected reserve network.
Oh, really?
Yeah, the genetic treasure was existing almost entirely on the private forestry land.
Wow.
If conservationists had just stuck to the national parks, they would have missed the most crucial genetic diversity of the entire species.
That completely flips the traditional idea of conservation on its head.
It really does.
And while this delicate diplomacy with timber companies is happening on the mainland out in the Pacific Ocean, there is a very different, much more aggressive battle happening.
Yes. If we look at the dispatch from the Island Conservation Alliance, working with the Chilean Forestry Corporation CONAF on the Juan Fernandez Archipelago.
Specifically, Alejandro Selkirk Island.
Exactly.
This island is a global biodiversity hotspot, packed with endemic species found nowhere else on Earth. But they aren't just building fences there. They're actively eradicating invaders.
Right, because island ecosystems are incredibly fragile. The native flora and fauna evolved over millions of years without many natural predators or aggressive competitors.
So when something new arrives,
When humans show up and introduce alien species, it is essentially a localized apocalypse.
wow.
The mandate on Alejandro Selkirk Island is aggressive, physical removal. They are targeting invasive plants like maquis, myrtle, and blackberry, which aggressively smother the native flora.
And they're also going after invasive mammals, right? Rabbits and mice?
Yes, which the dispatch notes are severely limiting the survival of ecologically important native species.
But how do you even go about getting rid of every single mouse on a rugged remote island without accidentally poisoning the native wildlife?
It requires extreme precision.
I imagine.
It involves mapping the entire island, doing manual trapping, and carefully timed baiting programs. They have to strike when the invasive populations are most vulnerable, but actively avoid the breeding seasons of native birds.
That sounds incredibly difficult.
It is an exhausting, cliffside, all-out effort to hit the reset button on that island's ecology.
So we've successfully navigated the corporate forestry land, eradicated the invasive species on the islands, and got these seeds to the airport.
Right.
But getting them to the UK is only half the battle.
Right.
Once that DHL air freight box lands, how do they actually stop the biological clock?
This is where we shift from the chaos of the field to the highly precise physics of the lab.
Okay, into the vault.
Exactly. Once those seeds arrive at the seed bank, it is a race against physiological aging. We have a highly technical workshop source strictly focused on the Brassicaceae family, which is the mustard family, but the rules apply to orthodox seeds across the board.
The objective is basically cryogenic freezing for plants.
Pretty much.
You have to slow down the aging process so the seed can tolerate sub-zero temperatures without dying.
Right.
And the physics of this are governed by two golden rules of thumb, according to the document.
Yes, very important rules.
Rule number 1. For every 1% decrease in seed moisture content, the storage life of the seed doubles.
Yep.
And rule number 2. For every 5.6 degrees Celsius decrease in temperature, the storage life doubles.
It's a compounding exponential effect.
That is wild math.
It is. The workshop source specifically notes that the optimal long-term storage conditions to preserve the physiological state for a model plant like Arabidopsis is bringing the seed moisture content down to 5% and then freezing them at minus 20 degrees Celsius.
I see a logical extreme here, though.
Oh.
If taking out water continuously doubles the lifespan, why not just put them in a tiny oven, bake them down to literally 0% moisture, and make them immortal?
That's a fair question, but biology is rarely a straight line.
Too good to be true, I suppose.
Exactly. This introduces the concept of equilibrium moisture content. Drying a seed isn't just about forcefully sucking the water out. It is about the seed finding equilibrium with the relative humidity of the air around it.
Okay.
And there is a hard biological floor to this process.
Meaning you can actually dry them out too much.
Completely. The source explicitly warns against ultra-dry storage, which is bringing the seed moisture below 3 or 4%.
What happens if you do?
If you bake them down to 0 or dry them too intensely, it causes severe internal damage to the cellular structure. Ultra-dry seeds actually deteriorate faster than those maintained at a slightly higher 5% moisture level.
Oh, wow. So it backfires.
Big time. So they have to use incredibly precise dry rooms set to 15% relative humidity at 15 degrees Celsius to slowly, safely coax the seeds down to that 5% sweet spot over about a month.
So you have these perfectly dried seeds frozen at minus 20 degrees. How do you actually know they are still alive?
Right, the monitoring phase.
Because if you look at them, they just look like tiny dead pebbles. You can't just plant them and wait a month to see if they sprout every time you want to check the inventory.
No, you definitely need faster answers. While they do run germination tests on small samples, scientists heavily rely on rapid biochemical assays.
Like what?
The most common is the TTC test. TTC is a specialized chemical salt. When you expose the embryo of a seed to TTC, if the cell is alive and actively metabolizing, it reacts with the chemical and turns a vivid, bright red.
Oh, that's cool.
And if it stays pale, the seed is dead.
It's a color-coded life detector. That is brilliant.
It is brilliant, but it contains a really tragic irony.
What's that?
The TTC test, along with other tests that measure how much a seed leaks amino acids to gauge cell membrane health, is entirely destructive.
Wait, to test if it's alive, you ruin it.
Yes. To definitively prove that a seed was perfectly viable and entirely capable of growing into a majestic tree,
You have to kill it.
you have to kill it in the testing process.
That is the ultimate scientific catch-22.
Hmm.
To know the seed survived the vault, you have to sacrifice it.
Sadly.
Which perfectly explains why they need to collect 20,000 seeds to begin with.
Exactly.
You need enough to bank, enough to share, and enough to sacrifice over the decades just to monitor the viability of the collection.
The scale of human cooperation, science, and sheer will required to do this for tens of thousands of species is just difficult to overstate.
It really is, and it brings us all the way back to the surface. It is incredible to look at the chain of custody we've just explored.
It's a long journey.
You start with a tiny, threatened Batavia punctata seed clinging to life in a fragmented Chilean forest on private timberland.
Right.
That seed is randomly sampled alongside thousands of others to capture 95% of its genetic code, complete with its soil pH and cultural history.
Yep.
It is documented, legally authorized through international benefit sharing laws, so no one is robbed, packs up, shipped across the ocean, slowly and precisely dried to exactly 5% moisture, and then locked in a vault at minus 20 degrees Celsius to essentially cheat time.
It is a meticulously designed archive of life. But, building an archive for the future raises one incredibly complex issue.
And that is what I want to leave you with today. The next time you walk through a botanic garden, or even just plant a seed in your own backyard, think about this massive, invisible infrastructure of law and science working quietly in the background to ensure these species exist tomorrow.
It's always happening behind the scenes.
But I also want you to consider this. We're freezing the genetic diversity of the year 2001 or 2026 in these sub-zero vaults. What happens when we finally thaw these seeds 100 or 200 years from now?
That's the big question.
With the rapid, unpredictable pace of climate change, will the specific soil, the weather patterns, and the exact pollinators that these seeds were perfectly adapted for even exist anymore? We started by talking about building Noah's Ark for plants. But the real question is, when the floodwaters finally recede, what kind of Earth will the Ark actually land on? Or are these brilliant seed banks simply building an archive for a world that has already disappeared?